My name is Shivik Narang, and my project is predicting the effect of novel missense mutations in neurological disorders on protein structure and function in silico. Now, when I was in 8th grade, I became really interested in learning about the brain. And as I read all about the brain, I learned about some diseases such as Alzheimer's, autism, and Parkinson's. And what I learned all about these neurological diseases is they all seem to have some genetic implication related to them. Alzheimer's had the APP protein in ApoE4 along with presenilin 1 and 2, while autism has over 100 genes implicated in its disorder. But other lesser known diseases, like the lissencephaly, in which the brains of individuals is not fully developed, causing them to have smoother brains with less surface area, leading to mental retardation and possibly even death, even those diseases had genetic causes. Now, in genetic causes, these are often caused by mutations, and there are multiple types of mutations. We have frameshift mutations, in which a nucleotide is added or removed, causing the entire DNA sequence to shift over. Insertions, where whole fragments of the gene are added, and deletions, where whole fragments of the gene are removed. We also have nonsense mutations, in which there's a premature stop codon, causing the gene to terminate early and the polypeptide cannot be produced. In all these cases, the polypeptide which is produced does not resemble the original structure in any way, so it's degraded. But there's another type of mutation, the missense mutation, in which one nucleotide is switched, causing a change in the amino acid. Now, while this may seem to not have as much of an effect as the degraded mutations, which is true, the missense mutations cause one amino acid to be substituted with another, and since amino acids have different properties, this causes a change in the folding of the protein, causing a change in the structure and possibly even the function. But understanding how the missense mutation causes the conformational change is a really hard problem to solve, and so that's my goal of my project, which is to find a quicker way to observe changes in the conformation of a protein based on missense mutations. Now, when I say quicker way, that implies there already is a method for figuring it out, and that's true. What happens currently in wet labs is that they first figure out where the mutation is, what the missense mutation is, but then it takes them around 6 months to later figure out how that mutation causes a change in protein structure and folding. And those 6 months, they conduct various wet lab tests and assays, and it's just really time consuming and really expensive because keep in mind, there's multiple mutations for a disease, and each of these mutations they have to conduct various tests to figure out how it causes a change in protein. So I wanted to use computer models, which is in silico, to see if I can make a prediction about the change in conformational structure, just without, in a much quicker and cost-effective way, without having to spend too much time. So for that, I first figured out 30 softwares online which have the potential to perhaps figure out a change in protein structure and to model the structure of the protein. And I ran them in my positive control group. Now, as you can see here, my positive control group consists of wild-type and mutant protein amino acid sequences. And I used all of these tests, Liz1, DCX, TTR, DNMT1, and contactin-associated protein, and these are all genes implicated in the formation of brain development, and when there's a mutation in these genes, it often leads to diseases such as lissencephaly, in which there's abnormal development. I checked all these mutations, for example, F31S, where phenylalanine at the 31st position is replaced by serine, and since this is my positive control group, these are mutations who are already known to be deleterious to the structure of the protein and whose effects on the protein structure had already been documented in the literature. So I ran my 30 protein softwares on this positive control group, and in the end, I came up with 5 softwares that were shown to work for all of these positive controls. So the 5 softwares that shown to work are shown here: MuPro, Provian, Polyfen2, Snap2, and iMutant 3.0. AIDA seemed to work, but eventually the errors were not consistent, so we had to get rid of that. Now let's quickly take a look at these softwares. So MuPro, it essentially indicates if there's a decrease in the stability of protein structure by calculating the thermodynamic stability, which is delta G. So in this case, since I'm using F31S as my model for representation, this shows there's a decrease in the protein structure. The next software, Provian, also predicts if a mutation is deleterious or not, using and calculating a score. Polyfen2 gives an extra layer showing if a mutation is probably damaging, possibly damaging, or benign, giving more support to the previous evidence. iMutant 3.0 classifies a mutation as disease-causing or not. And Snap2, which is probably one of the most important ones, it iterates over at the mutation point for all amino acids. So as you can see, F31S was the original mutation and is shown to have an effect. But not only does it show that what would happen if phenylalanine is replaced with serine, it shows what happens if phenylalanine is replaced with all 20 amino acids. And you can see in most cases there's an effect, but in some cases it's even neutral. So these are the softwares I used, and I then ran them on my experimental group. Now, my experimental group consisted of novel missense mutations. Now, what do I mean by novel missense mutations? A novel missense mutation is a mutation which is found to be deleterious, so the researchers already know it causes a disease, but but they haven't gotten around to figuring out how it causes a change in protein structure. And so that was the goal of what I was trying to see, if I could figure out how there's a change in protein structure in this experimental group. So I ran basically all the software on them, and here's my results of the experimental group. It shows it's decreasing deleterious, which is basically giving insight, okay, yes, these are causing a change in structure. And similarly, these 5 softwares also had for the positive control group run, and there were really high accurate percentages, over 95% for all of them, validating my softwares. So now that I've seen that, okay, this is deleterious, once again finding more support to the softwares, I still don't know how it causes a change in structure, right? Before I can go around to figuring that out, I just have to make sure I don't have any false positives in my softwares, right? So, for example, what if a software is just saying everything is decreasing, even though in some cases the mutation may not have any effect? So I ran them on a negative control group consisting of polymorphisms, which are known not to cause any disease, but it's just a mutation that's like a silent mutation, no effect on protein structure. But as you can see here, since there's MuPro is all red, which indicates false data, it was giving tons of false positives. So I had to get rid of MuPro, as that showed, just to give false positives. But now that I have my softwares, 4 softwares, and it's shown to work on the experimental group and the negative and positive controls, now I have to figure out how this causes a change in structure. And this comes to the iterations. So as done in SNAP2, for example, where phenylalanine is replaced with not only serine but all 20 amino acids, I did a similar step for 6 of the experimental group mutations for all 5 of the softwares, or now 4, because MuPro is giving false positives. So for example, in CNT-NAP1 C323R, which as you may remember is one of my experimental control group mutations, I replaced cysteine C with all 20 amino acids at the point, not just arginine. And so I did this for 6 of these experimental groups, and my results indicated that my hypothesis, which I could use in Silico software, was partially supported, since 4 out of 6 mutations, just using these 4 softwares, was able to give me an insight on how there's a change in protein structure and folding. So let's go to one of them, right? In C323R, only cysteine was shown not to be damaging. Replacing cysteine with anything else is damaging. And there's something interesting about cysteine. Besides cysteine, methionine is the only amino acid which has sulfur, and methionine often serves as a start codon. Therefore, cysteine is the only amino acid with sulfur, and it's basically solely responsible for forming disulfide bridges. So replacing cysteine in C323 would probably have an effect on a disulfide bridge. And interestingly, just very recently, there's an article that came out that showed C323 and C355 are involved in a disulfide bond. So this validated my result that C323 occurs because there's a change in disulfide bonding. Now, the next one is based on E493 in GAN1. So if we take a look right here, E493 in GAN1, it shows that there was a negative charge, right? Since we replaced glutamic acid with any other with a non-negative charge amino acid, it was shown not to work, though eventually it just may have been the presence of a charge that matters, since replacing it 2 of the positively charged amino acids do not seem to have an effect. So in this case, we can make it a conclusion that E493 is involved in electrostatic or ionic interactions, right? Now, for the S317N and KAF2A right here, it was shown that replacing serine with threonine and cysteine, which were other small hydrophilic molecules, did not seem to have an effect on the folding and structure of the protein, did not cause any deleterious effects. And this occurs maybe because they're all the same shape and same size, indicating that they're involved in a hydrogen bond which bigger molecules are not able to fit or accumulate. And then there was also the E128K from PAFA-H1B1 right here. And as you can see here, that was also involved in charge and electrostatic interactions. So once again, replacing that would affect an ionic interaction in the molecule. So my hypothesis is pretty much validated, right? So I was able to use 5 softwares, narrowing down from 30, and I was able to figure out why there's a change in structure of the folding of the protein for 4 out of 6 of them. And so this indicates that I might have to use a few more softwares to get a full picture of how there's a change in the conformation of protein, and that's what I'm working on right now, finding better softwares, including ones that can give models for my secondary and tertiary structure. Currently, all the models for secondary structure gave inaccurate data that did not match the literature, and for tertiary structure, they tried to basically fit the mutated folded protein onto the original wild type, therefore not showing an accurate depiction of how the mutated protein looks like. This project has relevant applications in the lab, as it can basically provide a much quicker and much more cost-effective way of understanding how there's a change in conformation of the protein, allowing researchers to devote their resources and focus on other tasks while getting a much quicker and accurate representation of why there's a change in the folding structure. Thank you so much.